Okay, so part one, first we're going to talk a little bit about what we refer to as parental gametes. So parental gametes are the allelic combinations on the gametes that were used to produce an individual. So I have a mother and a father, and I have a genotype. And so let's say I'm heterozygote for two different genes. So one way we could write um, that genotype is this, capital A, capital B, little b. And so there's actually two scenarios where one can become a heterozygote this way. So in the first scenario, your father gave you both of the dominant alleles. So on that gamete that was present within the sperm cell that led to fertilizing the egg, if we could peer into that gamete and see what exactly the allelic combinations are, we would see a capital A and a capital B. And then in the second situation, and then the second the homologous chromosome was provided by the mother. So she also has an egg, and within that egg there was a single copy of that same chromosome, and her combination was little a, little b. And so when we combine these two together, we have heterozygote copies of each gene. But we could also have the situation where my father provided the capital A, little b, and my mother provided the little a, big B. And so this is important in this chapter because of this idea of physical linkage. So in both situations, the genotype is the same, but in one case, the linkage is different. So in scenario one, the big A allele is linked to the big B allele, and the little a allele is linked to the little b allele, while in the second situation, that combination is the opposite. So this is what we refer to as the parental gametes, the combination of alleles that were present in the sperm and the egg that led to the fertilization for an individual. So this also motivates an alternative way to list a genotype. And so we use this designation whenever we are referring to genes on the same chromosome. So if we know that two genes are actually linked on the same chromosome, then we can use this, and this just tells us what was the combination in the parental gamete. So now we know that the capital A, capital B was the parental gamete, little a, little b was the other parental gamete, and then the opposite situation for the second scenario. So because recombination exists, because during prophase 1 the tetrad forms, and you actually have recombination between the two homologous chromosomes, we are not limited by the parental gametes. So I have the ability to link gametes in a way that was not present within my body. And that's because our body goes to this trouble of actually forming the tetrad and recombining between the homologous chromosomes. And so let's say we have a situation right here where we have a crossover between two of the chromatids. So these are non-sister chromatids right here because one comes from each of the homologous chromosomes. And so we have an exchange in the way you would see what the new gamete would look like is you just kind of follow from one side through the X to the other. And so now if we look at the result of this meiosis, you have one chromatid that did not undergo any sort of recombination. You have a second one that did not undergo any sort of recombination. And then we have two chromatids. So the first one that started here and ended here, that's this gamete right here. And then in the second situation, the one that started here and then moved on to here, that's what's shown in this gamete. And so now we can see that this particular meiosis has produced 4 different types of gametes. In the same way as all 4 different combinations. So whether or not a recombinant is formed depends on where exactly this recombination event has occurred. So you can imagine the recombination event might have occurred on the left side of gene A. In that case, we would not have seen these. But it's important that we're able to refer to these. And so these are either referred to as crossover gametes or more commonly, in this class at least, as recombinant gametes. So it's important to know when I say parental gametes what I mean. So these would be the parental gametes right here, and then the recombinant gametes would be the new combinations that are produced during crossover. And so this is just a continuation of a previous slide. So if you have independent assortment, where the genes are on different chromosomes, each combination is produced at the same amount. If you have linkage where crossover doesn't occur, then in this situation, you only produce the parental gametes. So none of the recombinant gametes are actually produced. However, in real life, you have crossover, and so now we have to deal with probabilities. And so if we look at, not just a single meiosis, but like thousands or millions of meioses, if we looked at, for example, 1 million sperm cells, and then we just calculated the frequency of the 4 different combinations. So, we would have 4 possibilities. And let's say we looked at 1 million sperm cells, and then for each one we just kind of tallied up. The first one was big A, big B. The second one was here. The second one was this. The third one was this. And we just continue to tally and continue to tally. We might get a number that looks something like 3,000, 1,000, 1,005, just to make it clear that they can be different numbers, and 3,054. Okay. So what's clear from this is that in real life, you don't have 25%, 25%, 25%, 25%. You don't have 50%, 0%, 0%, 50%, which are these two kind of like extreme approaches, but you have something in between. So the parental gametes are always the most common. So that's how you can recognize what the parental gametes are, is you look for what's most common. So we would say this is a parental gamete and this is a parental gamete. And this ranges between 50 and 100% of all the gametes that are produced. And then the recombinant gametes range between 0 and 50%. And so what determines that exact frequency? Well, it's actually the physical distance between the two genes. So consider on this left side the case where we have two genes, Y and W, and they are physically very close to each other. And then in the second case, we're looking at two genes, W and M, that are much further apart. And so, we can create a heterozygote individual. So this F1 individual is heterozygote right here. And then if we just look at the types of gametes that are produced, so the parental gametes would be YW, Y plus W plus. And so we can see in this situation, the two parental types are actually very common. So 99.5% of the gametes that are produced are the parentals, and only 0.5% are produced are the recombinants. But now in the second situation, W and M are physically very far apart from each other. So if we look at the number of base pairs that actually separate these two genes, it's much higher. And so in this situation, it's much easier to produce the recombinant gametes. And so the recombinant gamete frequency is actually 34.5% versus 65.5 parental gametes. So it's much more likely. And the reason for that is just the fact that there's a lot more places that a crossover can occur that would create the recombinant gametes. So in these two cases, we could do something like this just to show kind of randomly where they might occur. And so if we just draw 3 of these, only one of the 3 would result in a recombinant between Y and W. The crossovers here would have no effect on the combination right here. Whereas if they're physically far apart, only this one won't actually create recombinants between those two genes. So it's simply a probability matter. Like when you look at thousands and thousands of individual meioses, the location of each of these recombination events varies, and so the number that actually create recombinants between two genes is dependent upon the physical distance between them. And so this motivates this idea of map distance. So we want to be able to measure the distance two genes are. And so this is called a map unit, and the typical unit for these is centimorgan. So we call kind of in the same way you have kilograms, the way you refer to the distance between two genes for genetic distances in a unit called centimorgans. And each map unit represents a 1% recombination between two genes on a chromosomes. So in genes that are 50 map units apart or 50 centimorgans apart, crossing over can be expected between 100% of tetrads. All right, so let's look at two different scenarios. And so how do you actually do this? So the way you do this is you recognize the recombinant gametes, and you divide that by the total number of gametes that are produced. So scenario one, we have unlinked genes, so these are on different chromosomes, and we're looking in this heterozygote. And so we want to ask how many, how many of each gamete are produced, and in this situation, an equal likelihood of each gamete. So we have, 25% for each, 1/4 of each. So the number of recombinant gametes would just be adding up the two recombinant gamete frequencies, and you get a total of 50%. So the map distance, you wouldn't actually calculate this for genes that are unlinked. You would only calculate this for genes that are linked, but that would be approximately 50 centimorgans because 50% of the genes, or 50% of the gametes are recombinant. Now, in a second situation, we have a heterozygote individual, and so we know this is the parental combination because of the way we're writing the genotype. And so now, if we look at the gametes that are produced by this individual, 3/8 of them are parental, 3/8 of them are the other parental, 1/8 are one of the recombinant, and 1/8 is the other recombinant. And so if we add up the recombinants, 25% of the total gametes are recombinant gametes. And so this is simply what we need to use in order to calculate the map distance, and though this translates to a map distance of 25 centimorgans. So you always need to be thinking about gametes if you want to be calculating the map distance. Like, think about the gametes that are produced. All right, so an example of estimating genetic distance. So let's say I gave you information. Very typically, you'll see information that looks something like this. Where I tell you I took a heterozygote individual and I crossed this to a recessive, homozygous recessive individual. And then I observed the phenotype. So in the offspring of this, I looked at 1,000 different individuals, and I looked at their phenotypes in order to determine the genotypes of those offspring individuals. So, presumably this is, if it was plants, it would be something like, the height of the plant and the color of the pea pod. So, we can look at the 4 possibilities, and we would know this would be like a tall yellow pea pod, a tall green pea pod. This would be a short yellow pea pod and a short green pea pod. So we'd have all these individuals, the thousands of individuals we would phenotype, and then we would infer the genotype of that individual. And we can do that because this is a test cross, and a test cross, we know that this individual always gives the same gamete, little a, little b, little a, little b. And then there's 4 different gametes that can be produced by this parent. And so really by looking at the genotype of the offspring, what we're inferring is the gamete that is produced by this parent. So we know the second gamete is always this, and then the phenotype is always determined by this. And so by counting these, we now can say, okay, what is the parental combinations? So in this case, this and this are the 2 highest categories, and so we know that this and this was the original parental gamete combination in this individual right here. And then we also know that the recombinant gametes, these are the less frequent, are this and this right here. So now if we want to calculate map distance, now it's just straightforward. How many recombinant gametes are produced? 100 plus 100. How many total gametes are produced? So if we add up 400 plus 100 plus 100 plus 400, the total is 1,000. And so simply dividing the 200 by 1,000, we get an answer of 20%, which translates to 20 centimorgans or 20 map units. So this was, almost completely straight, almost exactly the same as this, but we also added the addition of a test cross. And the reason we add the addition of a test cross is that it's not actually easy. There's not an easy way to genotype of gamete. So if we look right here, and let's say we isolate the sperm, there's no easy way to know what is actually present within this sperm cell. Is it the capital A, capital B, capital A, little b? You think about this allele might control something like pea pod color, but a sperm cell is not going to have any sort of color associated with it. It's not like we can look at the sperm cell and say, oh, it's a yellow sperm or a green sperm. So there's really no way to infer what the actual genotype is. And so this is just an experimental trick that allows us to do that, because we've set it up in a very particular situation so that the phenotype of the offspring is determined by the genotype of the gamete. And so that's how we're able to actually calculate, the genotype of the resulting gametes. So this is an example of a single crossover, which is kind of what we've talked about so far. So this is a tetrad that is formed in prophase 1. And so you can see we're creating this new combination. And so, two of the non-sister chromatids undergo genetic exchange. 2 of the chromatids do not undergo exchange, and so 4 different types of gametes are formed. But you can also have a double crossover, so you're not actually limited to only having a single crossover event for a tetrad. So this is an example where 2 different crossovers have occurred, which straightforwardly is just referred to as a double crossover. And so you have 2 chromatids, again, that are not affected, so the exchange did not occur on these outer ones at all. But the non-sister chromatids have had 2 exchanges. And so how do you figure out what is produced? Well, you just kind of follow the pattern. So you follow it to this side, and then you kind of follow it back over here. So you have this orange, blue, orange pattern, and that's this thing right here. You could also have this following it from the blue, then you cross over to the orange, and then you cross back over to the blue, and that's where this gamete right here was formed. So just following where those crossovers occur, and then you exchange between the maternal and the paternal sources of the DNA. So you have non-crossover gametes, and now we have what's referred to as double crossover gametes. So this is going to become important for when we're starting to look at more than one gene at the same time. All right, so linkage in action. How does it affect ratios? So now we'll have a problem where we say A and B are 20 centimorgans apart, so I'm giving you the map distance between 2 genes. And so now we're going to have a little bit more complicated across, so we're going to have an intercross. And so what I want you to be able to answer, for example, is what percentage of offspring do you expect to have the recessive trait for both genes if you intercross AB, the AB heterozygotes. So again, we're specifying the linkage, the original linkage, and now we are doing, instead of a test cross, we're doing an intercross, and so you have to worry about map distance in both cases. So how do we do this? We do this using Punnett square, but now we're going to modify Punnett square a little bit to allow for the addition of probabilities. So, hopefully by now the way you're understanding Punnett squares is you have 2 individuals, and to create the square, each individual, the first thing you have to do is figure out the types of gametes that can be produced. So in this situation, 4 different gametes can be produced by both individuals. So they're both heterozygote, and so we first write the 4 types of gametes that can be produced. So big A, big B, little b, little a, big B, little a, little b. And so that individual goes there, and then we also do the same thing for the other individual on the other side. So this individual also can produce 4 different types of gametes. But now we have to worry about probabilities, because each of these gametes are not formed at the same rate. And so we have to use the map distance to actually figure out the probability of producing each of those gametes. And so, 20 centimorgans, so 20% of the total gametes are recombinant gametes. And so if we add up these 2 numbers, we see 0.1 plus 0.1 equals 20%. So this is where I got the frequencies by just using this map distance. And so I've just filled in the probabilities of producing each of those. So 10% plus 10% equals 20%. And then I just use the fact that they all must sum to 1 to figure out that the parental gametes is 40% and 40%. So I just change that to a ratio. So it's 0.4, 0.1, 0.1, and 0.4. And then the same is also true of the second individual. So 0.4, 0.1, 0.1, and 0.4. And so now when I fill in the square, I not only combine the genotypes of the gametes to get the genotypes of the individual, but I also multiply the probabilities together. So this is 0.16 because 0.4 times 0.4 is just 0.16. Here the frequency is 0.04 because 0.4 times 0.1 is 0.04. And then in some cases it's down to 0.01, 0.1 times 0.1. Okay. So I do this for the entire square, and now if I add up all of these numbers together, they're going to add up to 1 because the genotypes have to be they have to add up to 1. So the original question is, what percent of offspring do you have to expect to have recessive trait for both? And so the answer to this is just this part of the square. So this is the only individual that is recessive for both traits, and so the answer to that is 0.16. And so there's a 16% chance, or 16% of the individuals will be recessive. If I asked you, what percentage of individuals would be dominant for both traits, then I would have to group these together. So you would have to find all of the individuals that had the dominant traits for both. And you would just have to add each of those together. Okay, so that is the end of part 1, and next we will move into the situation where you have 3 total genes to worry about at the same time.